Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Good morning one and all. Welcome back to my channel Pari's tutorial. In this class I want to discuss about a separation technique which is used very wisely in in the laboratories, okay, for the separation of the macromolecules, biomolecules. So separation of DNA, separation of RNA, separation of proteins. Okay, so based on that, as in the last class we have already discussed about the process called centrifugation, where there's a separation of heavier particles from the lighter particles. But this time I want to concentrate on the topic called electrophoresis. Okay, so what is electrophoresis? The movement of charged molecules under the presence of electric field. See, each and every particle has got a charge in it when it is placed under electric field. So based on it, it moves towards the particular electrode in the opposite charges. For example, anions move towards the positively charged anode, and the cations, which are positively charged, move towards its opposite electrode called cathode. So this is the main basic principle involved in the electrophoresis. So how can the macromolecules or the biomolecules be separated in electrophoresis? Let's discuss in this class. Okay, so what is DNA and RNA has got a relatively a negative charge. So it is easy for the separation, as you can place it when it is placed under an electrode under electric field. It obviously moves towards your opposite charged electrode, right? In the same way, proteins are having both, like it could be either positively signed or it is negatively signed, because proteins are nothing but a chain of amino acids, right? So these chain of amino acids have to be placed under one, have to made be into one charge. So that can be done with SDS electrophoresis. So today in the class, let's talk about all the types of electrophoresis. Okay, so what are the types of electroforces that are involved? See, out of which STS is mainly involved for the protein separation, whereas agarose is mainly involved for the RNA and the DNA separation. So this STS being a vast topic, being a bigger topic to discuss, I want to talk first about STS page. Okay, so what is STS? STS is a sodium dodecyl sulfate polyacrylamide gel electrophoresis. What is sodium dodecyl sulfate? Sodium dodecyl sulfate is a anionic detergent. So what happens is, the protein sample generally have many bonds in it, like hydrogen bonds. It could be disulfide bonds. It have hydrophobic forces between the amino acids in the protein chain. Okay, so all have to be denatured and made it into a primary linear form, and should have a charge for the separation. For that to happen, we have to add the sodium dodecyl sulfate, which is an anionic detergent. So what happens is, for the electrophoresis to start, the sample which needs to be separated, that particular protein sample need to be added with STS, which is called an anionic detergent, and it is added with beta mercaptoethanol. This beta mercaptoethanol is meant for the breakage of disulfide bonds between the amino acids. So it is also added into the protein sample. Along with it, we need to add bromophenol blue. This bromophenol blue acts as a tracking dye that would help you to understand the bands when particular sample moves towards the gel when placed in electrodes, when placed in under electric field. Okay, so it is mainly, this separation mainly depends upon the molecular weight of the protein. You all know that higher molecular weight can't move faster, so they get they move slowly, and smaller particle, smaller weight molecular proteins would move faster. So this is the thing that we have in the sample. So what does actually the process, what is the apparatus of STS? So there are two glass plates involved. So I want to discuss it better in this thing, so that it would be easy for you to understand. So what really happens, and how it, you know how the separation happens. See, this is your electrode that I'm talking. For example, this is your, these are the two glass plates, okay, out of which they pour the stacking gel and dissolving gel. There are two different types of gels in SDS page. Okay, I will tell you the pH and what is the buffer that is used in SDS page also. So what they do is they take two glass plates and they are separated with a spacer in the middle, okay, and they are meant to have a small spacer so that gel can be poured. Now first of all, first gel poured as dissolving gel, and later it is stacked with a stacking gel at the top. So once both the gels are poured into the glass plates, you will be placing a comb, okay. This particular comb will form wells for you where you can upload the sample, okay. This is the whole thing. It takes time for you to at room temperature, it takes time for you to fit, like the whole comb has to fit into the gel. After some time you have to remove the comb so that the wells have well framed for the sample to load, okay. In meanwhile, once all the samples that have been added into it, all the things that you have added, even you will add glycerol even to that, because glycerol will help you to increase the density of the sample, okay, along with bromophenol blue, along with SDS, along with the beta mercaptoethanol, along with the protein sample, you will be adding the glycerol also for increasing the molecular weight of the protein so that it won't mix with the running buffer. It would be in the it would be inside the well itself, okay. So all the samples have to be heated first before you upload, before you load them into the wells, okay. So for that you have to heat it at 95 degrees centigrade. At this heat, the bromophenol blue, sorry, the SDS would and that anionic detergent would attach particularly to the protein molecule, making the protein a complete negatively charged. All the disulfide bonds, all the hydrogen bonds, all the hydrophobic forces are being denatured and damaged in the protein, okay. And the whole protein has now become ready, and it has completely changed to have a negative charge, so it is ready for the separation based on the molecular weight, okay. So this is the I'm talking about. This is pH of 6.8 and it is a pH of 8.8. See, this is just I'm drawing a, this is the comb wells for the separation of your, right. I'm okay. Now once these are all ready, your sample is ready, okay, your sample is ready and you got everything in it. And first of all you have to add with the basic standard what you call a standard protein molecular weight ladder, okay. So that ladder will give you a bands, okay, that you will be adding in one of the wells for your comparison. At what band it's a molecular weight, like this should be higher kilodaltons, should be lower kilodaltons, okay. It is measured in kilodalton, okay. So this is your molecular weight, standard molecular weight ladder for your comparison. So let's think this is sample 1, this is your sample 2, this is your sample 3, okay. So let's think in that way. So what they do is once the sample is ready, you will be placing this in a buffer. SDS will be placed in a buffer and you will add a running buffer. What is the buffer that is used is tris glycine chloride. So glycine plays a very important role in your SDS page, because glycine, I will talk to you why glycine plays a very important role in SDS page, because based on the pH the glycine's charge keeps changing, okay. It's general, it has a charge of C. This is the glycine molecule that I am talking about, right. This is the glycine at pH normal, it is normal, like if it is neutral, if it is at normal, if it is at neutral pH, this would be a normal charge, it doesn't have any plus or minus. So based on the pH, alkaline pH or acidic pH, this particular glycine keeps changing. It could be either positive or it could be either negative. Okay, that is the main principle involved in SDS. So chloride ions would always have a minus charge. So chloride ions would definitely go towards your opposite charge, right? So it always goes toward your oppositely charged electrode. Okay, electrode. Okay, see now. So this is your yeah, this is your tank. Everything is ready. You have set samples ready. You have your stacking gel and this ready. Okay, these all the samples move towards your opposite direction. So all are now negatively charged. Okay, they have to move towards your opposite electrode, right? So here what happens is, when there is a passage of, when it is placed for electrophoresis, when it is under the presence of current, the sample starts moving. So in the samples got your, when the buffer passes through it, right? When buffer runs through it, the samples contain all the glycine. See, when it enters into the stacking gel, it contains glycine molecules at the top, okay, with proteins to be separated in the middle, and you have chloride ions, because chloride ions move faster than glycine, because glycine molecules, when they enter into this particular stacking gel, the pH is 6.8. So because of this, they can't move faster, because see, it's a 6.8, it's almost neutral, so it doesn't have that particular charge, so they move little slower than chloride ions. So chloride ions move faster and they reach the stacking, they reach this particular stacking end of stacking gel. In this end, when they reach the stacking gel, all will be at one position. So chloride ions at the base, with proteins to be separated in the middle, and with the glycine ions in the top. So chloride ions move faster, you all know that, right? Glycine, they take time, and proteins to be separated are first made to have a linear position, because if this doesn't occur, all the protein molecules will have different movements, and that is why we get non-specific bands. When when you stain them, you don't get specific bands, right? So they have to be first of all made into a linear way. All the protein samples should have should start at one starting point, right? So for them to make linear, these glycine molecules and chlorine molecules would sandwich them, right? Like they will glycine molecules from the top will push the protein molecules down. At the same time, chlorine molecules will forcefully make the protein molecules move down. So here all the three things would come into a linear stand. All the protein molecules, glycine, everything will be linear. So now once they enter into the resolving gel, it's also called separating gel. Okay, this is the main gel where separation happens. Okay, so once it enters, the glycine molecule now enters into the alkalinity of alkalinity, right? 8.8 is a alkaline pH, right? It's so now the glycine molecules would move faster. What happens is, these chloride ions would move faster, but the these glycine molecules, when they enter into the resolving gel, they gain movement and move faster and moves towards your opposite electrode. So they will cross the protein molecules to be separated and get settled at the bottom. So based on it, all the chloride ions and glycine ions would come faster, leaving all the proteins based on their molecular weight separation. Okay, larger molecules would be at the top and smaller molecules at the top, that you already knew based on their molecular weight. So till stacking gel, all was stacked up. All the three were stacked in a linear position. Once the resolving gel has started, because of the change in pH, the glycine and the chlorine would go faster, leaving the protein bands at the back. So finally we'll get good protein bands. Now these bands, once the whole thing is done, we'll switch off it and we'll take the gel out. Is done, we'll switch off it and we'll take the gel out. And we don't need this stacking gel, we only need this resolving gel. So we take this particular resolving gel Okay, this, for example, this thing, this is the resolving gel. Okay, so this based on already you have added the basic protein molecular weight ladder, right? Like this is the for your comparison. So based on it, for example, this is your sample 1, this is your, so this is the sample 2 and this is the sample 3. Okay, so we've added the samples into the well, into the wells, and we got the gel. So based on the known molecular weight of the protein ladder that we have added as a marker, we'll compare the molecular weights of the unknown proteins. Okay, and the molecular weight of these proteins are measured in kilodaltons. So the higher kilodalton weight would be in the first band, and the lower kilodalton weight of the protein molecules would be at the lower band. So based on it, we can compare the unknown samples. So once the gels are obtained, okay, once the gels that have been obtained, we have to go for the staining technique. So you all know what is staining technique. Staining is meant to visualize the protein bands better. Okay, so the gels that have been bands that have been obtained in the gels can be visualized better when they are added with a staining solution. So what is staining solution? Staining solution mainly contains about Kumasi brilliant blue, methanol, acetic acid and water in particular ratio. Okay, this particular Kumasi blue would help you to visualize the protein bands better. So the whole gel has to be placed in the staining solution. Make sure it is completely immersed in it, and it should be placed in a shaker for 1 hour at room temperature. Okay, so due to this, the whole bands would be visualized. Then after that, you have to again destain the gel, destain by placing back to a destaining solution. This destaining solution mainly contains only methanol, acetic acid and water. Doesn't have any Kumasi brilliant blue in it. Okay, this destaining would help you for the gel, the to removal of that particular color which is not attached to the protein bands. So it will get dipped out of the gel because of this destaining. Okay, so this is the apparatus about SDS. How in lab we organize SDS electroforces in this diagram. Okay, so I told you these are the two glass plates. They are placed in the have two type of gels you have and everything. Okay, see, let's summarize it as simple as possible through this diagram. Okay, the sample is placed added with SDS and all, and they will reduce, they will denature them. Okay, the whole protein molecules have been denatured, and you have got your linear form of proteins, and now they are all loaded with the proper with the proper gel casted. Okay, and then you will get your samples, and you will get everything placed under current. Okay, so this is the whole mechanism of SDS. So till now we have discussed about SDS, and in the next class I will tell about the agrogel electrophoresis, which is meant for the separation of DNA. Okay, so okay, hope you like the class. Hope you are hope you got a good detailed understanding about this process. Okay, so see you in the next class with the interesting topic. So thank you, have a nice day. Please subscribe, like and comment about this class. Thank you.